Hello, good morning, good afternoon. We're a minute early, but we can go ahead and start our seminar. Welcome. Please let us know where you're joining from. It's October 30th. This is the third presentation for our 10th International Plant Breeding Seminar Series, hosted by NC State University here in Raleigh. We have the honor today of having with us Dr. Tom Poorten. Tom earned his PhD from the University of California in Berkeley, where he cultivated a passion for translating genomic research into tangible results. He worked as a postdoctoral fellow at the strawberry breeding program at UC Davis, where he assembled a draft octoploid genome and built bioinformatic pipelines to as a base for gene discovery and marker assisted strategies. Today, he's a senior bioinformatics scientist at Pairwise here in the Research Triangle. And Tom builds genomic resources and bioinformatic tools for gene discovery, allele design, and vector design. His work supports the company CRISPR-based crop editing platform and helps the gene editing team accelerate the creation of improved crop varieties. Tom, it is a pleasure to have you with us. I'm going to stop sharing and give you the floor for you to start your presentation.
Great, thank you, Carlos. Okay, sharing my screen should be coming up any second. There we go.
Yeah, it looks good, just presentation mode. You're good.
Yeah, thanks, Carlos. Really appreciate the opportunity to talk at this seminar series. So yeah, we'll jump right in to this. Just a brief agenda to talk about. So I'll start with a pairwise overview, and then I'll do a short section on the blackberry industry and breeding history, and then go through our gene discovery experiments for thornlessness, and then we'll talk about actually the gene editing process for thornlessness in an elite line, an elite cultivar. And I think we'll have time for this last section and talking about a few other traits that we're doing gene editing for. Just quickly about myself, that was a nice introduction. This map, I couldn't help it. This is from the books and show Game of Thrones. It's not a real map, but my career path has kind of done a crisscross pattern across the United States at these different universities. So been experienced with different types of academic settings, and then yet my most relevant, as you mentioned, experience was at UC Davis at that strawberry breeding program there, developing genomic resources for the octoploid strawberry. And now I've been at Pairwise for over 6 years, continuing this similar type of work, developing genomic resources, often for non-model species. And then I'm also involved with gene discovery projects as well. This picture is just from last weekend. We were at a pumpkin patch in North Carolina and went over to the thornless blackberry you pick, where you can come out in the summer and pick blackberries, and it was neat to see some thornless blackberries growing there at that farm. So what is Pairwise? We're a technology-focused food and agriculture company that makes tiny changes to plant genetics to solve big problems. We're based in North Carolina for the most part. We have our headquarters in downtown Durham with office and lab space. We have a second site about 15 minutes down the road in Research Triangle Park, where we have our larger greenhouses, growth facilities, and then more lab and office space. We do have people spread around the country, and in particular, of course, out of people out in California, which is a real hub for the berry industry in the United States. And for the past several years, we've been building out more and more collaboration. So I just want to touch on that in the next couple slides. So we're currently on our second deal with Bayer. Our first 5-year deal, this was a collaboration to work on row crops, as you might expect. And our second one is focused on short stature corn. On the other side of the slide, Corteva, we're also working with another of the larger agriculture companies to do gene editing in their row crops. In the middle, this is our collaborator our commercial partner for the caneberry program. So that commercial partner is Plant Sciences Genetics. They're based out in California, and this includes our work on blackberries and raspberries. And I'll talk about this more throughout the presentation. And just to mention a couple other collaborations that are a little bit more recent. So we're working with the IITA on improving yam. Um, and this is funded by the Gates Foundation. For this, the main trait that we're focused on is reducing the stature, going for a shorter stature yam to reduce labor, environmental impact, and enabling mechanized farming. Hopefully that's the goal there. And then with the CIMMYT group, this is a case where we're out licensing our CRISPR tools for them to use in their wide range of crops, and we're excited to see what they're going to do with it with the CRISPR tools that we've developed internally. And next, I'll talk about just a kind of a quick overview of CRISPR and gene editing for people who might not be too familiar. And we kind of have this analogy of modifying sentences. So there's three main types of CRISPR gene editing. And this first type, we'll talk about traditional CRISPR nucleases. And here, what is happening is you end up with a short indel. So in this analogy, we're removing a word. And one of the key parts about CRISPR, and this is throughout the case, all three cases, is we're able to zoom in, to home in on the exact location in the genome to make these precise changes. So in the second case, base editing, this is when we're changing, by analogy, a misspelled word. We're changing just a single letter or just a short string of letters. And then finally, templated editing, we're finding a misspelled word in the genome and changing that entire word. So swapping something out that we don't want and swapping in something that we do. And now I want to talk about how we go about finding gene targets for gene editing. This is, of course, contrasting to what a lot of people are familiar with marker-assisted breeding. You have a QTL, you can develop a marker and deploy that in your program. With gene editing, of course, you need to go that step further and try to find that exact gene target, and hopefully even that exact mutation that could be segregating in the population. So we have these two main approaches that I'll talk about briefly here. So comparative inference on the left, this is just the example where we know something about a trait-associated genes from another crop, and we could try to translate over to our crop of interest. So we find an ortholog and deploy that in a gene editing pipeline. With this, you can think of it as having a medium probability of generating the desired trait, and it will likely require more shots on goal. So trying different edits, or possibly trying different genes. With this other category, the gene discovery in the focal crop, and that's what I'm going to talk about in the coming up with this discovering the thornless gene, is when you have a trait segregating in a population, we can design, execute gene discovery experiments, and then hopefully identify the causative mutation. And once that happens, we have a higher probability of generating that desired trait because we're still working in that same crop. On the flip side, this can be challenging. Some alleles are just recalcitrant to discovery. It's just hard to find that right gene in your gene list or find the right mutation. I'll go through this, what we call our pairwise gene editing pipeline. And most of these steps correspond to the functional groups in our pairwise R&D group. So starting with a trait, these first set of teams work together to identify gene targets, and then we want to zoom in. Do we want to edit the promoter, for example, or do we want to edit the coding region? Make those decisions. We work with what we call our tools team. These are the experts in molecular biology, biochemistry, to actually pick the right CRISPR enzyme to use and for a particular edit. We program the editing tool by designing the particular guide RNA, and this will allow us to, again, target a very specific part of the genome. These can come together into our editing tool constructs and is handed off to our transformation team. Often this is a very difficult process. Some crops are harder than others, and blackberry was one of those cases where it was more difficult and took time to develop those transformation protocols. Once we do get gene editing out of transformation and the regeneration process, we go through the molecular screening process. For this, we do um, deep amplicon sequencing, and we're looking through all of our plants to find the plants that we have our desired changes. And then those are handed off into the controlled environment, into the trait testing teams, and we do the characterization for performance. And I'll just call out some of the key players who are involved with the gene discovery work here for thornlessness and blackberry. So Brian Crawford, Brian St. Albin, and Gina Pham played some key roles in the research projects that I'll talk about. Before that, just a quick um, run through the blackberry industry and a little bit of breeding history. So the current global market size is roughly $1.5 billion. In that plot on the top right, you can see this has been growing fairly steadily over the past few decades. These different lines are showing um, volumes by origin. The red line is just the sum. But then below that, you can see the blue line, um, the, the highest location is Mexico. They're the largest producer of blackberries, and then below that, the next set of lines are from California, Central and Southern California. Also trying to represent that in the map on the bottom right. In terms of the U.S. market, you can see again, like Mexico provides a huge portion of blackberries into the country. Again, with California, and then also the southeast of the United States being another location. And then in the northwest of the U.S., I have that star in Oregon. This is mostly for the processing part of blackberry industry. So these blackberries are produced mainly for things like frozen berries or jams. And then just in terms of predicted growth, this 4% is the expectation or prediction for the next decade, this compounding annual growth rate. So expected to keep growing. So yeah, introducing the trait here a little bit more. So thorns, or you could call them prickles as well, they're problematic for caneberry production. They can slow down production, or they can increase production costs due to a slower harvest rate, and they're also a safety issue for workers. And there's been a real trend over the past several decades for growers to really prefer these varieties that are thornless. Um, However, these thorns are still, or the alleles for thornlessness are still present in the genetics of a lot of the commercial caneberries. And then lastly on this slide, just to talk about nomenclature, if we're being really careful about botanical language here, prickle would be the right term to use. And in our manuscript that we're working on for this work, we're going to use that term, but for the sake of convenience and familiarity, we're going to use the term thorns in this talk. However, if you do a little bit more reading for raspberries, spines is another word that comes up as well. But again, we'll just try to make it easier on ourselves and use thorns. So I introduced a little bit more about blackberry cultivar development here and some of the key traits. So I'll talk through the traits first. So there's 3 types of growth habits: trailing, semi-erect, and erect. These trailing varieties, these tend to be higher ploidy, hexaploid, octoploid, and higher. And these tend to be in the Pacific Northwest for that processing industry. The erects or semi-erects, these are our varieties that are mainly used for the fresh market. And then in terms of thornlessness, in the table you can see that there's several different sources of thornlessness. We're going to focus on this one Burbank / Merton thornless. I'll talk about that more soon. And then the last trait, primocane flowering. So typically, caneberries will grow in their first year, they're called primicanes. Their first set of canes are called primicanes, and they won't make flowers and fruit. They'll go through winter, and on the second year, they'll put out fruiting laterals. Those are called floricanes, and that's where the flowers and fruit will come from. But there's recent development of primocane flowering, so these cultivars are able to produce flowers and fruit on their first year growth. And the University of Arkansas program has been successful at deploying that into several different varieties. So next to talk through more about the different sources of thornlessness. So it goes back 100 years. Thornless evergreen was one that came out of Oregon. Merton thornless, the second one here from 1956. This came out of a breeding program in England. Austin thornless was found 1961 in Texas. Lincoln Logan, this one, Logan berries is a hybrid of blackberries and raspberries. And the Lincoln Logan was a thornless variety discovered in New Zealand. And then lastly, raspberries, this Glen series from Scotland. Some of these are thornless, might call them spineless. So all these are different sources of thornlessness in caneberries. This initial one that I mentioned, thornless evergreen, turned out to be not very useful for breeding because it was actually a chimera. So when breeders were trying to use this in crosses, it wasn't genetically inherited. So this led to more the search for more sources for true thornlessness. And just to focus on the Merton thornless and derived cultivars from that source. So this, I'll mention this is a recessive allele, but this has been used successfully in several different programs in the United States in particular. So the USDA ARS Beltsfield program in collaboration with state partners, starting in the 1960s, developed these semi-erect thornless varieties listed there on through the decades. University of Arkansas also has been successfully using this Merton thornless allele in their breeding program, first with Navajo in 1988 as their first erect thornless variety, continuing to use this allele in subsequent releases. And NC State as well has used this allele in their variety called Vaughn about a decade ago. And one more thing that was really interesting, a colleague, Brian Crawford, found in the literature going back through some of these older papers on thornlessness. So the Scottish breeder Derek Jennings reported in 1985 when he was talking about Merton thornless, and he mentioned in this kind of brief review how it was later realized that the Merton allele was the same as the one designated as this S allele in raspberries. So this got us thinking as we we're kind of going through our genomic data for both blackberry and raspberries, we should be looking out for this. And our last slide here in this section, to talk about how this was mapped previously by linkage mapping. Just a little over a decade ago, a linkage map was made for this mapping population, this in tetraploid blackberry. They were using 119 SSRs, and they found the QTL on their linkage group 4. So I'm going to turn over to the next section here, going through our gene discovery experiments. And first, I'll talk about our resource development. So this was several years ago now, a collaborative effort to develop better resources, both genotype and phenotype diversity data sets. So pairwise along with these partners, our commercial partner, Plant Sciences Genetics, and then these public entities, USDA, NC State, University of Arkansas, Cornell, University of British Columbia. So bringing some of the important caneberry breeders from North America to put these resources together. These reference genomes were this was more of a project from one of our collaborators, Margaret Worthington, that pairwise contributed a little bit to. But I'll mention these are 2 diploid genomes. This first one, Hillquist, so yeah, 2 diploid blackberry genomes, I should say. This one was released a couple years ago, and the second one, Rubus omifolius, Burbank thornless. This is in prep in conjunction with our paper where we're talking about these gene discovery experiments. These were developed with PacBio long-read sequencing, as well as Hi-C Illumina sequencing for scaffolding, and then expression data sets, RNA-seq, and then long-read ISO-seq to help us with annotation. And then building out really good genotyping data sets. Given that the genome size is fairly small, we're able to just do whole genome sequencing to roughly 30x coverage for a large set of accessions, over 600 USDA and public accession sequence. This does include wild varieties as well as cultivars. And then in terms of phenotyping, this was done in field conditions. So roughly half of those accessions were put in a field and assayed across these 4 trait categories: architecture, yield, berry traits, biochemical, and flavor, and then a little bit of disease sampling as it came up. And this diversity data set was, able to enabled us to look at the diversity of things like this, like trichomes and thorn traits across the whole rubus phylogeny. So this is spanning raspberries, and within rubus, raspberries and blackberries are fairly distantly related. And just scanning around, you can see that there's a diversity of trichome, density, presence or absence of thorns. And something that we noticed here, there has been speculation about correlation between trichomes and prickles. I'm using that word, the term prickles, but the same thing as thorns here. Um, but when we looked at this in this data set, we were noticing there was a large correlation between the presence of short granular trichomes and prickles. And so this got us thinking about the possibility of being developmental or genetic ties, connections between trichomes and thorns. So I'll get into our first experiment, so a genome-wide association study. This was a fairly small, but it was really targeted set of samples. So we looked through pedigrees of blackberry accessions and pulled out 43 accessions, so 29 thornless and 14 thorny. So we used that for initially available diploid reference genome, Hillquist, as the reference genome for this. And that reference genome will be used for most of the analyses that I'll show. So this was a SNP-based assay. So we had over 300,000 SNPs and used this GWAS poly R package and using a recessive tetraploid model. These are all tetraploid blackberries. And what we found was this large peak on chromosome 4 that corresponded to the linkage mapping paper that I mentioned earlier, Castro et al. And then also it corresponded to the same region found by this other group, Johns et al. Paper earlier this year that was doing a similar work in parallel as us. So this was a fairly large region, but we wanted to narrow it down further. We're trying to nail down our gene and our causative mutation. So we're trying to do more experiments here to narrow down the region. So the first thing the first type of project after the GWAS was a bulk segregant analysis type of study. So we had a heterozygous parent. Based on its pedigree, we thought it would have the thornless allele in its background. So after a selfing and looking at the offspring, we found this distribution of phenotypes between thornless and thorny offspring. And then with the bulk segregant analysis type of approach, we take leaves from our set of thornless offspring, so 32, and then 65 thorny accessions, extract DNA. And then from those 2 different categories, those are bulked together and sequenced to high coverage. And that's what I'm showing here is the results of that. And so we're looking for allele frequency differences between our bulks. So in the second track here, we're looking at the entire length of chromosome 4. And the bulk segregant analysis results confirm this peak in chromosome 4. This set of tracks below that is zoomed in to the end of chromosome 4, about 6 megabases. And this gray bar here is actually showing the eventually gene that we discovered underlying the trait. And these green and red arrows, you'll see this in the next couple of slides. I'm using these to show what our best guess at the narrowest version of our QTL would be. So going from GWAS to bulk segregant analysis, our confidence was raised that it's in this narrower region. So like I said, we wanted to do more to narrow down the region further if possible to get a shorter gene list. So here I'm just showing, again, the shorter, the zoomed in stretch at the end of chromosome 4. You can still see the GWAS and bulk segregant results there. And now I'm showing these identity by descent results. So this is cultivar by cultivar type of analysis. So we do a sliding window analysis of SNPs along the chromosome, and we're looking for regions of low heterozygosity and matching alleles. So these are just a couple examples, but Primark Freedom turned out to be the one that was most useful for us, given that it had recombination that broke up linkages that allowed us to further narrow down the region, at least a little bit in this case. And that's what we're seeing here with these green and red arrows is showing where we are confident that the Q-tail is narrowed down to. This slide has a lot going on, but we're bringing in now the red raspberries. As I mentioned earlier, there's speculation or a prediction that there's actually the same gene underlying thornlessness in blackberries and red raspberries. So when we look at our red raspberries, in this case, we're using a publicly available red raspberry genome to align the reads and look at SNPs. And we have these two thornless examples here, Glen Ample, Glen Rosa. These were just basically just to confirm that we still that we do have correlation in this region between the results here and the trait. But then in this example here, Glen Magna, this was actually thorny. We see this stretch of high homozygosity. That's what these lines bunched up here are indicating. So this is indicating to us that there is recombination that can break up the linkage here, and it allows us to further narrow down the region. And in the synteny plot over here, I'm showing the most narrowed down regions that we have. And we're trying to put information together from our different genomes. The top two are diploid blackberries. So Rubus argutus Hillquist, Ulmifolius Burbank thornless. These are diploid blackberry reference genomes. And then we have our red raspberry and black raspberry. This is the oldest one. It's kind of just been useful for a long time and maintain a lot of our analyses. And here in this green box, I'm showing the location of our gene that we found. I'll talk about that more soon. But just to talk about what's going on up here, we have this kind of interesting pattern. These V's or these bands crisscrossing each other. This happens when you have a tandem duplication in a genome, but we actually think this could be a misassembly in this genome, given that there's only one copy in Burbank thornless and in these other genomes. Yeah, so in this case, it's really this narrowed down region from this green arrow to this green arrow for that genome, and then this gene here. But at this point, we've narrowed it down to 400 kilobases, and we're starting to look. Sorry, now I added my animation here just to show how using the synteny plot, we have to kind of translate from one genome to the other, and we're going back to our blackberry reference genome to get our most narrowed region here. And we did one more thing here to try to not narrow down the region, but to try to help us rank gene candidates. So we did some RNA-seq with a set of samples from our bulk segregate population. So we had 3 thornless offspring and 9 thorny. And this plot it's a volcano plot with the p-values on the y-axis and then the expression fold changes on the x-axis with these higher values associated with higher expression in thorny versus thornless. So sometimes this differential expression in this type of context could just result from downstream effects of a mutation, but it's also the case that you can have what we are hypothesizing is happening here in this eventual gene that we found, WOX1, is that with a truncated, with a premature stop codon that led to nonsense mediated decay and lowered mRNA abundance, and this would have the impact of differential expression in this analysis. So really this was, we're jumping ahead, but it was a real process looking through our narrowed down gene list, looking through the functional annotation, looking at possible mutations, looking at possible expression hypotheses. But when we found this WOX1 and looked at the mutations, especially in our Burbank thornless, we found some interesting things. So on the top, this is our gene model for WOX1, the wild type version with 4 coding exons in those black boxes. And then the blue boxes are some of the key motifs in the protein. So this homeodomain motif and then this Wuschel box protein. And then when we look at the Burbank thornless genome and its alleles at this gene, we see this 2 different mutations in the 2 different alleles. So first, there's we're calling WOX1-1. This is corresponding to this 8 base pair insertion, and it's right in one of these key domains. And it's 8 base pairs, which means it leads to a frame shift mutation that in turn leads to a premature stop codon. So this could either lead to the nonsense mediated decay, like I mentioned, reduction in mRNA abundance, or if it goes through the process of translation, you'll end up with a truncated protein. With this other allele from Burbank thornless, WOX1-2, this is another out of frame mutation, so a 4 base pair insertion, WOX1-2 here in orange. And this one is also overlapping this one of the key motifs from this gene. And then this third mutation that we're showing is from Rubus idea, so that's red raspberry. And what's the name for this older cultivar that's thought to be the source, burn at home, the so-called S allele. So when you look at that, we see another 8 base pair insertion, which is another frame shift leading to a premature stop codon right at the end of that exon. And then when we start to think about how these are mapped out on to genomes, or think about how the genotypes look in these different cultivars. So Burbank thornless, as I mentioned, heterozygous, has these 2 separate alleles that could explain the thornlessness. And then look at a cultivar called Ponca, and you look at its genome at this locus, you could find that it has actually 4 copies of the WOX1-1 allele. We could also write it as this lowercase s, so quadruplex homozygous for that allele. Just a few other examples here. The thorny cultivar Primark 45, looking at this locus in that cultivar, it has 2 copies of this WOX1-1 allele, but it has 2 copies of the wild type allele. And this would explain why it's still thorny. And then lastly, this cultivar, this is coming from our commercial partner, Plant Sciences. So we're just calling it PWD760. This is our line that we're using in gene editing. And then looking at this gene copy, it has 3 copies of the thornless allele, WOX1-1, and 1 copy of the wild type allele. So when we design our gene editing, it's actually pretty convenient. We only need to edit 1 of the copies that is still wild type at this allele. At this point, it's all hypothesized, right? But we'll show you the results soon. But just to talk a little bit more about how this allele is present in the germplasm. So this WOX1-1, the main takeaway here is that this copy seems to be the primary source of introgress thornless trait in blackberries. This bottom cluster is showing a few different more recent thornless blackberry lines, and this allele, the 1-1 allele, are all clustered together down here. The second allele, 1-2 from Burbank thornless, wasn't appearing in the modern thornless germplasm. The rest of this tree is showing 2 other clusters. So this is the wild type blackberry alleles at this gene, WOX1, and then this cluster up here are red raspberry and black raspberry copies, more distantly related as expected. Now we want to just show how it looks in a larger context of the WOX gene tree, as the WOX gene family. So here we have some a wider scope of species, so Arabidopsis, these 2 diploid blackberry species, red raspberry, as well as Prunus avium, just to kind of give it more context in the rose family. And in this green box here, this is our WOX1 gene that we've been focused on. And in this little cluster down here, they're all very closely related, and there's no corresponding copies from prunus. And except for these copies over here, down here, with these Arabidopsis WOX1 genes down here. And also thinking about how it's reported, the reported functions of different Arabidopsis genes across the family. This WOX1 probably makes the most sense in terms of reported function. This is just to summarize the steps we did to narrow down the QTL, going from our initial GWAS, bulk segregant analysis work, and then the identity by descent analysis, narrowing it down further and further to the set of genes that we then carefully look through in terms of functional annotation, inspecting mutation, and then incorporating the transcriptome expression data as well. So with this, we thought we had high confidence as a candidate, and we wanted to move forward with gene editing in this line. I mentioned earlier, this is blackberries are not the easiest thing to work with in terms of transformation. So just to call out, this was a bit of a journey, as people here at Pairwise say. We have a great team of transformation scientists, and they made it work. But now we have multiple stable transgenic systems developed, as well as non-transgenic editing strategies that we were able to deploy. And in tandem, there's been years of editing tool development, so refining our CRISPR enzymes and so on. And finally, when we were able to get this to work, get the gene editing and get the plants out of the transformation process, it was exciting to see how our edited lines are now lacking the thorns as we were hoping for. And going back to the beginning in terms of trichomes, we also see a lack of glandular trichomes as seen in other myrrh and thornless derived varieties. However, they do maintain their simple trichomes. But it did seem to confirm that connection, developmental connection between glandular trichomes and thorns, or lack of thorns. This image at the top of the screen is showing how we were able to put our edits pretty close to the native, we're calling our native thornless allele. This is the 8 base pair deletion near that homeobox motif, and then we're able to put a deletion just downstream of it. And this likely led to a similar outcome in terms of a premature, well, it did lead to a premature stop codon and non-functional protein. So that wraps up the thornless gene discovery work. And okay, not a lot of time to go through this, but I'll go through this pretty quickly. This was an exciting trait as well that we're developing in blackberry. So it's not truly seedless, but we're going for seedlessness, similar to what you have in other crops like grape, where it's effectively seedless. So we're able to edit a particular gene target and then end up with this reduction in lignin in the endocarp. And I think this video should work, where it just squishes this seed. Yeah, there you go. So this is using a texture analyzer to analyze the amount of force required to crush the seeds, and we see an 80 to 90% reduction in force compared to controls. And these edited lines occur in field trials. Hopefully, in the years to come, we'll see these commercially available. Okay. Talk about one more aspect here in the next series of slides. So introducing it, this gene terminal flower 1. This has been a well-known gene for a long time. In Arabidopsis, TFL1 mutants exhibit earlier flowering time with less vegetative growth and a shift to reproductive development earlier. So earlier flowering, obviously of interest to a lot of crops, including blackberry. However, in tomatoes, when mutations of TFL1 ortholog self-pruning occur, this leads to a determinate rather than indeterminate growth habit. So we just see a change in plant architecture. So as we deployed editing strategy for TFL1 in blackberry, I'll just kind of go through this quickly. But so yeah, basically we found the ortholog in blackberry, and then we're able to generate these edits in the tetraploid blackberry, looking for all out of frame edits. to knock out this gene effectively. And then what we see in what we're seeing is a couple different things. So events that have 4 edited alleles of TFL, they have earlier lateral break and faster termination of branches with the terminal flower. And they have their first open flowers developing 7 to 12 weeks after plugging. They also have the shorter stature, so they have these edited lines at the bottom at 10 weeks and 20 weeks compared to the unedited controls at the top. So you can see, well, it's hard to see the flower. There are a few flowers at this 10 weeks and more early developing fruit at this 20 weeks, and some pink flowers are in the middle that you might be able to see, as well as the shorter stature is apparent between these two different, between controls and the edited lines there. And this is just building it out more to more data in the greenhouse. So we're seeing earlier ripe fruit development compared to controls in the greenhouse. And then this is being worked on more in terms of field work. And so we're trying out this new strategy compared to what is typically done with erect varieties like this, where you have this kind of unedited normal strategy of 3 feet by 8 feet spacing. With this more compact plant architecture, we're testing out this different spacing, reduced spacing, 1.5 feet between plants and 5 feet just between rows. And we have these field trials going on in California, as well as pre-commercial grower trials in Colombia, and then working on several grower, packer, shipper grower trials next year and the next couple of years in California and Colombia. And yeah, this is just wrapping up everything. So I think I'm right on time. So yeah, hopefully we're showing here gene editing offers a fast targeted strategy for improving crop performance. And it's a real complement to traditional plant breeding strategies. And here we're, been using a lot of genomics and molecular biology knowledge from other plants and utilizing that for gene editing and developing valuable products for growers and consumers. And that's basically what Pairwise is all about. So yeah, we'll wrap right there. I'm happy to answer any questions.
Thank you so much, Tom. Very, very interesting presentation. I ask people to post questions in the chat, please. If you wanna stop sharing, and we.
Sure.
I do waiting for questions to appear in the chat. I have particularly one question in the gene discovery process for the thornlessness gene. Did you consider looking at rose, the work that has been done in rose or other rosacea? Because I imagine there's quite a bit of work done in thornless rose.
Yeah, we definitely looked at it.
Okay.
And I think we need to go back in terms of go back to the literature at this point. That was at the early stages and seeing if there's anything that might translate to blackberry. But that's a good call out, yeah, to see
because I mean
what's
I
the
think that
latest there.
industry
Yeah.
is also very interested in thornless. They may
Yep.
be more readily available, I guess, in the genetic base because nowadays you don't see many thorned roses. But yeah, I thought about that because I grew up when my mom collected roses, and I grew up with my hands all hurting from roses. Okay, so we have question from Nancy. Knocking out WOX1 does bring an impact in yield or flavor, kind of secondary consequences of that? Blocking that gene?
Yeah, good question. Yeah, certainly looking at that, any kind of pleiotropic effects in greenhouses or field trials. So far, I don't from what I'm aware of, I'm not aware of any secondary effects. And again, this is the big upside when we're basically recreating a mutation that's in the gene pool already.
Okay. Yeah, that was one of my questions too, because I've seen in other crops very simple mutations directed at one trait, and then when we look at the gene-edited versions in the field, it was like, wow, all over for other traits. It
Yeah.
depends on which pathway that particular gene is. I mean, if it is a common pathway for other traits, then you really screw up a lot of things.
Yeah, no, it's certainly common, right? There's a lot of trial and error across our different crops and strategies. Pleiotropy does happen, of course, yeah, for sure.
Hmm.
In this
Yeah,
particular case, yeah, it's um
yeah.
yeah, the
Question.
mutation is in the
Yeah,
gene pool and we're able to kind of recreate it and hopefully
yeah.
minimize the effect. Yeah.
Okay, Don Jones asks if are blackberries primarily produced under greenhouse conditions or plastic house or inside or in field?
Yeah, I think hoop houses, I should have said that. The images at the end,
Yeah,
those were covered hoop houses, and I think
hoop
that's
house.
kind of become a typical way to do it.
Mm-hmm. 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 Gina Velasco is asking what was the biggest challenge you faced when analyzing the data or performing the bioinformatics analysis?
Good question. Yeah. Yeah. So one thing that definitely comes to mind is making when we're trying to nail down the gene, having really good functional annotation of the genome. It's worth putting extra effort in there. Um, you might end up with different types of functional annotation tables that you have to combine, and you have to be really careful with that. Um, because yeah, I think in this first iteration, we didn't. It wasn't very. Walks 1 wasn't labeled very correctly. Basically, unfortunately, this is kind of goes hand in hand with working with non-model species, and you just have to be careful at every step.
Okay. Jan is asking what was the motivation for going GWAS first and then moving to BSA-seq, since the BSA-seq can be done with bulk sequencing genotyping and give an arrow or entire region, would you only
Yeah,
do BSA-seq in the future?
yeah, that's definitely a good point about the strategy. Yeah, I maybe yeah, I should say we had this large project. We're looking at lots of different traits, and the GWAS was just there to do fairly easily because we were collecting all this genomic information and all this different trait information. So at that point, the easiest strategy was to do a GWAS. But yeah, you're right. GWAS strategy definitely can be the right thing to do if you're kind of narrow down in terms of your traits already.
Please keep posting questions in the chat. I have a time frame question. From the moment you guys started with this project until you got your materials in the being tested for production, what was the time frame?
Yeah, I don't have a it's this has been uh I want to say at least 5 years in terms of like starting
Mm-hmm.
the Rubus diversity project. Um yeah, it definitely takes a lot of time to do those types of field work, trials multiple and doing it for a couple of years.
Yeah. Yeah, but
But yeah,
once you do
if we
once you
were to
do
do it
this,
again, we would go
yeah,
faster, I'm sure. Yeah.
yeah, sure. But once you have it, I mean, then
Yeah.
next round of genetics will not need to will not depend on that early discovery phase again.
Yeah.
Nicolas Rueda is asking, let me see, the Ohio State University first. He's from Ohio State. First, thank you for the seminar. My question is, have you identified any temperature dependent flowering genes in rubus species? And do you see potential for using CRISPR or other gene editing tools to modulate their expression to optimize flowering in tropical climates like Colombia?
Right. That's a yeah, good question. At the moment, we're just seeing how TFL is working out. That's something that is talked about a lot, but we're not actively going after anything else at the moment. Yeah, definitely excited about how the TFL ended up um, opening up new avenues in terms of like the compact stature and hopefully improving yield. But temperature, yeah, can definitely have an impact, and that's something that we're watching out for in different locations.
Okay. So you're testing this in Colombia. You said Mexico is the main production area. Are there any restrictions about testing gene edited crops in Mexico or not?
Yeah, at the moment, my understanding is that yeah, growing gene edited crops in Mexico is not allowed, basically.
Okay,
Yeah, well, that's my understanding at least.
okay.
So yeah, that is part of the reason why Colombia and South American countries were considered instead.
All right, Gina is asking a question to us if she'll be able to access the recording. Gina, tomorrow you will receive an announcement for next week's seminar, and at the bottom of that announcement, you should have a link to the recording of this conference. Okay. Let us know if you have any problems accessing that link or playing the video, and we can get back to you. Okay. Is there a seasonal length of flowering affected by the early flowering trait?
Yeah, I don't have a great answer to that. That's something that collecting data on. Um, so yeah, yeah, we're just starting to collect data. So something like that would probably take a couple of years to nail down.
I know this is a question that you may want to answer or not. Nicolas is want to find out where in Colombia you're working, but I mean that's
Yeah, I don't know. Anyways,
yeah.
off the top of my head,
Okay.
anyways. Um,
Yeah.
I mean, I think if you follow us on LinkedIn, I think there has been some posts related to Colombia, but yeah, I'm not sure any details right
Mhm.
now.
Yeah. All right. Any other questions before? There's a lot of praise for your presentation, Tom, in the comments. But what is unique about Pairwise platform for editing?
Yeah, I didn't talk about too much detail, but we are. I mean, I think we have great people on the different teams. The transformation team, our gene editing technology, our tools team, really great people.
Mhm.
We have our what we're calling our Fulcrum platform, and we're starting to out license um, some of our own invented CRISPR enzymes called Shark, or kind of offshoots of Shark. And yes, a few companies are starting to out license that, as well as some academic institutions. So if you're interested in that, yeah, you can reach out to me and I can put you in touch with
Sure.
our business development folks.
Yeah, I happen to bring students to Pairwise, part of one of my courses, and it's always very, very interesting. And by the way, the Blackberry work is centerpiece for those visits. All right, let me share my screen again here, and thank you, Tom, for a brilliant presentation. And next week, we are going to have Dr. Hiwan Demisi from Ethiopia, Hawassa University, talking about breeding orphan crops at the same time, 11 a.m. Eastern time. Thank you so much for your attendance. Thank you, Brandon, for your help. I hope you guys have a great weekend and look for the announcement tomorrow, a week ahead or 6 days ahead of the next seminar. Thank you, Tom, again. It's been a pleasure. I hope to overlap with you in one of my visits to Pairwise sometime, okay? Say hi to you.
Sounds
All right.
great. Thanks.
Thank you so much. Everybody have a great weekend, okay? Bye-bye.